going to talk about the process of skeletal muscle contraction. Contraction starts with the action potential that's sent from the brain or the spinal cord and arrives at the axon terminal of the neuromuscular junction. When it arrives at the neuromuscular junction, it causes calcium channels to open, flooding the axon terminal with calcium. The calcium binds to the vesicles that contain acetylcholine, causing the excitosis of acetylcholine into the synaptic cleft, represented here. Acetylcholine binds to acetylcholine receptors on the motor end plate, which causes sodium to flood fibers and potassium to flood outwards into the synaptic cleft. This triggers a depolarization of the motor end plate, and it triggers an action potential in the sarcolemma as the voltage-gated sodium channels open, represented here. Afterwards, acetylcholine is broken down by acetylcholine esterase, represented by these little green guys here. The action potential is carried through the T-tubules, where it triggers a release of calcium from the terminal cisternae of the sarcoplasmic reticulum. Calcium binds to troponin, which shifts tropomyosin off the active binding sites on the actin. So the tropomyosin are these blue lines here, and troponin are the yellow little heads here on the actin, which are these purple circles here. Myosin heads use the hydrolysis of ATP and ADP to be cocked and ready to attach to actin once the binding sites are exposed. Myosin heads bind to the exposed actin and pull the thin filament, causing the sarcomere to contract. This is a representation of the rowing motion of the myosin heads that cause retraction. They remain bound until another ATP is hydrolyzed, which provides the energy to release the actin and return to their cocked position, represented here. Finally, the muscle relaxes when calcium is transported back into the sarcoplasmic reticulum. And that is skeletal muscle contraction.